Well, hello everyone. Welcome to Girls in STEAM and our micro medical panel. It's great that you could all join us today. My name is Brian. I'm joining you actually from Science World. So you notice it's kind of dark in the background here. I'm up on the top level of Science World, hidden in one of the corners of the OmniMax Theater, if you've ever been up here. I wanted to begin by gratefully acknowledging Science World is located on the traditional and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh First Nations. We're very grateful for the opportunity to work and play in these lands. Throughout the session today, if you have any questions, please feel free to type them in the YouTube chat over on the side. We'll try and get to as many as possible for our wonderful mentors that we have. We ask that you keep them relevant and on topic with what is being discussed. If you've missed any of the sessions or if you've come in a little bit later, feel free. All of them will be recorded. You can check them out as we go. This, again, is the micro medical panel as we go through. I think right now, let's just take an opportunity to meet our panelists. So our first one is Maya. Hi, Maya. Thanks for being on the show.
Nice to be here.
So tell us your name, what you do, and what is one thing you love about what you do?
So my name is Maya. My pronouns are she, her, and I'm a graduate student right now, which means that I graduated high school and then I did a 4-year undergraduate degree, and now I'm doing a 2 to 3 year graduate degree to get my master's. And one thing that I love about what I do is I think the problem solving is my favorite part. I love to think really hard about something and find a solution. And when you do find a solution to a problem, it feels really, really good.
And endorphin eureka rush.
Yeah.
What is the field for your graduate work? What are you specifically studying?
It's cancer research.
Okay, wonderful. I'd like to also introduce Elaine. Elaine, thanks for being with us. Or what is it that you do?
So I am the lab manager for the Advanced Microscopy Facility at the University of Victoria.
Oh my goodness.
And our lab is an open lab, so I work with everyone on campus plus off campus. And I do a lot of outreach programs in schools.
Okay.
And so I was trained as a marine biologist and biological oceanographer. And when I was doing my PhD, I got into electron microscopy. So marine biology is cool, but electron microscopy is way cool. And I work with everyone on campus, including fine arts and all the medical people. And fine arts wanted to know what was in their pigments. And so we have X-ray analysis on our scanning electron microscope, so we could tell them.
That is so cool.
My job is so cool. So it's an adventure every day. And whatever specimens come in, when it's the first time you've looked at, say, a beetle foot, somebody else may have looked at it. It may be published, but if it's the first time you look, then it's very exciting.
That is fun.
I've heard those, and it's really fun.
Yeah. Everything that Maya said about problem solving and the like is science. But mine is STEAM. So it's science, technology, engineering, arts, math, and biology. So the STEM words should be biology. And I guess that's in the part. Yeah.
STEAMBA. We've got it all coming through.
There you go.
Yeah.
It is. It's a STEAMBA. Yeah.
Just for those who are joining us, we do have a third panelist, and it's the wonderful world of being online. She is actually, there was a bus delay. She's trying to join us from her phone, but will be coming in a little bit later on. So the magical world of technology happening that way. For, again, those who are watching online, feel free to add questions in the chat. You can even add comments in there. I know we've got people visiting on our panelists from a few different areas, Victoria, Vancouver. I'm up in the Science World Dome. If you want to share where you're viewing from, we'd love to see that as well. It came up a little bit in your introductions, and I'm curious to hear a bit more about your journey to get to the role you are now, like where you thought you would be starting and what made some of these left turns and things that we had. Maya, did you always imagine that you would be going into this kind of medical cancer research field?
I think so. I think I had the interest in high school. I did some research papers or just like essays when I was in my science classes, like biology, chemistry, physics.
Right.
I didn't like physics, but I loved biology. So I went into biochemistry because I was very interested in the molecular world. So
Okay.
how little molecules fit together. And then during my undergrad, I studied a lot of proteins, and those make up a large portion of your body, and they do a lot of the work that keeps you alive. And I was really interested in how those reactions happened. And then I got interested in cancer because, it affects a lot of people, unfortunately. And in terms of a scientific problem, it's one of our biggest ones, and I really wanted to help contribute to that and figure things out.
Awesome. I apologize, Maya. At the very beginning of your sentence, things froze up a little bit on our side. I don't know if that happened for you as well, Elaine. But the topic. So is it cancer research is the one that you were most interested in then?
Yeah, currently.
Fun. I saw you smiling a bit while Elaine was talking about the coolness of electron microscopy and such. Do you get to use that in your field?
Not in my field, but during my undergraduate degree, they let you do a lot of different things, which help you decide what you want to specialize in if you do go into grad school. And one of the things that we got to look at in one of the courses was electron microscopy. And I got to bring in one of my flowers from home and put it under the little microscope. And I got to see what pollen looked like underneath the electron microscope, and it was very, very cool.
Fun. So Elaine, you started in marine biology. What took this left turn into what you're doing now?
So when I was 14, I was going to be a marine biologist. And boy, motivation is a wonderful thing. I found out I had to have French and I had to have this and that and the next thing. And suddenly my marks in those classes went up. Motivation. So I got into university. I did an undergraduate in marine biology and zoology. And there I discovered that if you want to do marine biology, you can come into it from any field. Or because then in the graduate, I got married. I had 2 boys, and then I was teaching in a high school biology, and then I found out that the school where I was teaching in, the principal had a heart attack in the middle of my first term there, and didn't come back till the middle of the second term, and nobody noticed his absence. The discipline was all what you made of it. I was lucky in biology because you could talk about the body as the human body, and you could, there was an empathy with the students who maybe had problems, but all the time, at one class, there's a hierarchy in a class, and these are high school students, and somebody at the back of the class had passed wind. It was awful. And he was a lower class kid, and the high up kids wanted to bash him up. But it was an immediate way of getting the kids involved, because I'm saying to them, okay, this could happen to any of you. So how would you prevent it? And suddenly we're into the bacteria in the gut and the bacteria that prevent passing smells onto the rest of it. And suddenly you're there with them, and it was exciting. But then I realized this was the school my kids would go to, and my class had 3 disruptive elements in it, and one of them had just come back from having been laid off or so. So I didn't want them to go to this school, and they wanted to go to the school I wanted them to go to. So that's when I went to do into the university and do a master's, so I could afford a school where I could send them. And I realized that the research part, and in the UK, you can do a master's in 1 year. It's extremely intensive. But I realized the research part of it, you do a 4-month research part. And I realized there that I liked research. Maybe I should do the PhD. But I wanted to do it on a part-time basis. So eventually, and because it's a hugely cost it didn't cost, like if it was $4,000, it was $400 to do it part-time. So
Gotcha.
it was, everybody wanted to do it, but you had to have a really good excuse. Well, having kids in school happened to be a really good excuse, and I got to do it part-time, which meant that I dropped the kids at school at 8:30. I was in the lab by 9. I had to leave the lab at 3 to pick them up. And it worked out fine because the kids went to my parents for the summer holidays. So I got to be full-time and so on. Anyway, it all worked out. I really enjoyed the master's, got to do a PhD. And it was in the PhD I got into electron microscopy.
Yeah. Now, I confess I don't know 100% about that. So if you want to look at something like a beetle foot, what's involved in electron microscopy imaging that? Like, what are the steps you would go through?
So what Hitachi have just given me a tabletop scanning electron microscope to take into schools.
Okay.
This was just pre-COVID. And then COVID struck and we couldn't go into schools. So fortunately, we live in the digital age and we have the internet and we can have Zoom on the microscope, which means now the classes can come to me. And so we have this program. The teacher just has to get hold of me and arrange a time. And now I can talk to schools in remote areas, which is great because I have the gift that keeps on giving as a specimen. It turns out that eelgrass is covered in diatoms, and diatoms are beautiful. The art in science comes into diatoms, but we also have ones of human body, and I have a stub of walking through a forest, so we have slime molds and fungi and ferns and stuff on this stuff. And when you look closely with an electron microscope, boy, it is beautiful. So and then wait. Oh, so this. See, yeah, and I needed people to know about it. Well, if I went through the school board, it would get stuck on the principal's desk because they're too busy. But so I wanted to go through the parents and the grandparents and the teachers. So I got interviewed on North by Northwest about this program, and I got a lot of schools come at me through grandparents and parents and teachers, and it works really well. So I'm hoping that anybody out there would like to have a play on a scanning electron microscope. And the ideal thing about this tabletop, we have an $8 million microscope in the lab. Well, it started off at 8 million, so it's a few more now. And it looks at the picometer scale. So if you think millimeter and divide it by 1,000, you have a millimeter. A millimeter divided by 1,000 is a micrometer. And a bacterium is 1 to 5 micrometers. Then if you have the micrometer and divide it by 1,000, you have a nanometer. And a virus is about 30 to 100 nanometers. Now you take the nanometer and divide it by 1,000, you have a picometer. But you've seen one atom. You've seen them all. And the tabletop scanning electron microscope may not have the resolution of the picometer range one, but it's still a lot of fun because now you can put anything you like in it, and it will take wet stuff. So you can take a leaf and put it straight in. If you want to do other electron microscopy, you have to process it.
Right.
You put a grape in a vacuum, you'll get a raisin eventually. So cells have to be processed properly, but not in the tabletop. It's so much fun.
Well, looking at this is our micro-medical panel, so kind of looking at the microscopic layer of things. Maya, you mentioned a little bit you were looking at proteins and things. What other factors on the micro level kind of into your work, things that are very, very tiny that you're trying to research or work with?
So jumping off what Elaine had been saying, I think a lot of what I do is imaging based. I use the microscope almost every day, and there's different microscopes that I use. I study stem cells and stem cells, so I want to know why a normal stem cell becomes cancer. We know that it does, but we don't know the steps in between. So I do things to sort of look at the in-between and find out what are the steps that get something normal to become cancerous. And I use a lot of microscopes to image cells and groups of cells. So I'm always looking at cells. I once called myself like a fancy photographer with a science degree because I took so many photos.
Yeah.
And we also look at cells after they've been sliced in like a cross section. So you preserve the cells in a wax, and then you slice it really thinly, and then you can look at that under the microscope and see the nucleus, and sometimes you catch cells dividing, which is quite fun.
That's fun.
And there's a lot of interesting stains that we can put on. Maybe some of the students are familiar with like gram staining bacteria, where you find the ones that are pink and the ones that are purple, and you know a bit more about the types of bacteria that you're staining. I do that with my cells, but instead of gram staining, I'm staining for specific proteins. Maybe they're associated with cancer, maybe they're not. Just trying to figure out what's going on at the cellular level.
That is so cool, Maya. That is wonderful. And that is a leading technology. I mean, you're at the leading edge of some really important work that's coming up now. That's wonderful.
I feel like with science, you always feel like you're on the cutting edge of something. And I think that's one of the most exciting parts about being in STEAM.
Yeah.
Just this feeling you're maybe looking at something that no one else has seen before.
It's like the new age exploring. You can't really explore land as much as we basically know the planet, but we don't know the molecular world as well. And it's really exciting to be able to figure out what's going on at that level.
What are some of the hurdles you faced in your career? Elaine.
So I wanted to do, when I was in my PhD, I wanted to do electron microscopy, and Southampton, UK, where I was, didn't have, the oceanography department didn't have an electron microscope. And so I had to go to physiology to use theirs. And I met this guy who had a small facility, one guy on his own, and he says, oh, it was like hitting a brick wall. You don't know anything. I haven't got time to teach you. It's much too expensive. And it was a brick wall. And then I talked to a colleague in oceanography, and they said, well, why don't you go to the general hospital? And the guy who ran the facility was my professor's research assistant 10 years before. So anybody coming from oceanography was made very welcome. And it was wonderful. It was an open facility. And there were 4 technicians, and they all decided I was going to learn electron microscopy. And it was brilliant. Oh, this is easy. And they made it as easy as possible. And I got hooked, like really hooked on it. And so my facility is run in the same way. It's easy. Come in, I'll show you how to do it. And we train our users mostly. We will do the work for you, but we'll charge you for it. But if you want to learn, you can learn. And I've got undergraduates coming in doing directed studies and teaching them to use the electron microscope and making them independent. And then they get it on their CV that they can use an electron microscope of one kind or another. And so, that's fun. So if you can think outside the box and go in a different road, it's not always a straight road, it's a different road. You'll be so much better off. Yeah. And just because I had a husband and kids and I wanted to go in one way, you just have to work out how to do it. It's not difficult in the end. Well, there are, you have to be able to manage time a little bit, but worth it. Totally worth it.
Maya, for you, what were some of the hurdles you faced so far?
So I just basically finished my first 6 months of grad school when the pandemic started. So it's been really hard. And a lot of times master's degrees are supposed to take, 2 years. I'm finishing my 3rd year in September, and I, fingers crossed, will be done by then. But having my project stopped and having to go home and not do anything was really stressful, and it was really difficult to go in like the end of March and kill all of my cells. That was a very drama-filled day for me, and having to sort of start back up again when there wasn't a lot of people that were allowed in the lab. And my lab is really, really big. So we had a lot of people who were all trying to come in at the same time. And that was a bit hard. Sometimes you wouldn't be able to go in. And I rely a lot on what the hospital does to do my work. And they weren't working as much because they were focused on COVID. So I had a lot of technical issues doing my science. And then towards the end, when I did all of my experiments and I finished them, we had a bit of a product shortage. So it was just like one thing after another. And our result wasn't that significant. So after all of that work, we didn't really find anything, which is a result in itself. That just means that our hypothesis was wrong. And that happens. And I sort of have to live with that.
Maya, I got my PhD on that. Because when I got my external examiner, everybody said, "Oh, you got who? You poor thing." Because he had a reputation of really being bad. But what he liked was, because I had tried a whole bunch of things that didn't work, but I had put those results in my appendix. He liked that. Not that everything worked perfectly. So you need to keep that.
Yeah. We're going to publish what we found because it is significant. The route that we thought the cell takes to get to cancer isn't the one that it actually takes. So it is something else. And now the next person can take on that question and solve it.
We have a question coming in from our viewers out here. They're wondering, actually, I'll pop it up for you here. What are some of the soft skills you find that are useful in your profession? So not so much the technical, but the human interpersonal ones. Elaine?
Oh, you just have to be in my lab. I want the lab to be open, and I would get third world country men come in, and they see a woman and they don't want to talk to me. Well, when it first happened, I would chat with them because they couldn't go to anybody else. I was it at the time. And so eventually they stopped thinking of me as a woman and started thinking of me as somebody who could actually get them some results. And once that switch happened, it was good. Now, it used to take, 10, 15 minutes or so of chatting with them before I saw that switch on their faces. Now I've got it down to about 5 minutes, and it works so much better. So those communication skills are kind of make it easier, let's say.
Do you find there are techniques that help bring it from that 15 minutes down to that 5 minutes? Any magic you can share with others?
Well, only getting them to talk about their research. Because once they start talking about their research and what it is they need, and that I can help them get there, then and it's fascinating because I get to see a lot of different people's research at the front end and before they've actually published, because images are so cool. Yeah, I've just got big cameras.
Maya, what are some soft skills that are helpful on your end?
I would also stress that communication is really important. You meet a lot of people, and there's a lot of strong personalities I found throughout my career and my time in school. And you just sort of, like any workplace, you have to manage your expectations, learn how to get along. And just, I found that what helped me was just being a nice, personable person who values compassion, and that has helped me along the way. You're not always going to meet people that are going to like you, but that's just life, and that happens wherever you work. That happened to me when I worked at a grocery store while I was in school. And some other soft skills I think are important is, I think historically science has been very closed door from the outside world. Sometimes we're so focused on our little problem that we don't see the bigger picture. And one of the things I do is I study a woman's cancer. So I study cancer that happens in the uterus, and I've been doing a lot of advocacy work in a little side project about how maybe we should be more inclusive, because there are people who are non-binary or transgender that also have this cancer, and we're trying to make the research and the healthcare itself more open to people, to make it more inclusive. So I'm really lucky my supervisor lets me do advocacy work as a part of my degree, and also lets me do outreach. And I feel like those are important to me.
Just like being taken care with the language that's being used, that sort of thing.
Yeah, and just making sure that like I'm thinking about the greater social and cultural aspects of my project. So I'm not stuck in my little box of science, and I know that there's more out there.
I
Yes.
think you've got it. You stuck it right, Maya, because you found a supervisor that you get on with, and in a subject that you're passionate about. And I think that's what, when you go into undergraduates, you get the chance to check out some of these professors when you go into their lectures. And if one of them is doing something that you and can communicate what they're doing passionately, and it's something that you want to do, those are the ones you have to go after. And as an undergraduate, I found that the undergraduates who go up to to a person and say, I really want to work in your lab and I'll do it for free, I'll volunteer in your lab, when they get to get graduate school, they will have that experience, and boy, they will be a commodity that a professor will want, because they've got that, and they've given up their time to get it, but it has worked. It'll work wonders on their CV.
That's exactly what I did. I think sometimes people worry, or I did when I was, I think, in high school and in undergrad, that my grades weren't good enough. I'm not the best on at tests, but I was one of those students that asked all my professors, can I work in your lab? Can I work in your lab? I'll do it for free. And I did. And that's what got me into grad school. It was those experiences. And one professor, he was like, I took a chance on you and it really paid off. And yeah, so marks aren't everything. And if you're thinking about going into a science or STEAM degree, it's a lot of exploration. You get to try a lot of different subjects to find one that you're interested in. So it's very exploratory that way.
Awesome. Great question. Thank you. Anyone else who has questions in the chat, we'd love to hear some more of those. It's kind of come up a bit here. I want to make it more formal. Our viewers today are sort of in the 14 to 16-year-old range. So imagine you are talking to a 14 to 16-year-old Elaine or Maya. What information would you give that person? Elaine?
I think that's really what it is. We've just actually discussed this. If you go into university on one line, don't worry if you find that you want to go in a different direction, because universities are flexible enough that you can change majors quite reasonably easy, especially if you found a professor that you would really like to work with. Go for it. Yeah, do exactly what Maya did. Go say, please, can I work in your lab? It will give you on your CV experience. And when it comes to employing somebody, experience counts a lot more than does marks.
Maya, what would you tell a 14 to 16-year-old Maya?
Yeah, I think if you know that you're interested in science and you're thinking of pursuing it, I think you just have to sit with yourself, and if it is something you decide, like, you just have to make the most of it. I really, really enjoyed my undergraduate degree. I took a lot of different courses, some in science, some not in science, and I think about them all the time. I think when I was 14, that was about 10 years ago, so it's been a while. But I think if you're passionate about something, you should definitely go for it. It's worth all the hard work. There was blood, sweat, and tears involved, but I think overall it was just an amazing experience. And if you're interested in these fields, you get to explore a lot of them. And even if you don't end up doing science after, I think all of those experiences are really fun. And in the world that we're in now, you can really there's a concept called like interdisciplinary research, where you get to combine, maybe science and art, or science and economics, and you can really go a bit farther than you would have a long time ago. So there's a lot that you can do, and if there's a problem that you see, and you're passionate about it, that's where you should be.
Wonderful. I have some exciting news. Our third panelist has made it. I'm just going to bring him in now. Hello. I know it was an exciting day for you. Hi.
I'm
Could you?
sorry for being late. It's just
No
late
worries.
stuff. Anyway.
Could you let us know your name, what you do, and we've actually just been chatting a bit about advice you might give a 14-year-old you. So if you're
Sure.
looking back.
Yes. Hi everyone, I'm Vaishnavi. I go by the pronoun she, her, and hers. I'm currently a PhD candidate in cell biology at UBC. I think Maya is also at UBC, if I'm not mistaken. Yeah. I'm hoping to finish up soon and studying something about how different parts of a cell they communicate with each other. And advice I would give to a 14-year-old self is, I mean, honestly, like, I did set my heart on being a scientist at that age, but I didn't know a lot of things that would come with it. So don't be bogged down. I mean, no, at that stage, you're like, oh, scientist, I want to be a scientist, but I don't know if I can reach that level. Or, they're like, I would see if this is a pyramid, they were like at the top of the pyramid, like the star of a Christmas tree or something like that. So I used to feel like, I don't know if I'm good enough for that, and I think a lot of us, we feel that, but I think you've gotten already amazing advice from the other speakers that if you have the perseverance and the dedication, the interest, and the right people to help you along, actually. So we didn't have a lot of mentorship cafes like this, but I was lucky to have people to help out also. So yeah. I just feel, just be passionate and just stick to your goal, and it's fine if there's some detours on the way.
For everybody, I think, what would be your dream project, your dream piece of research that you would like to work on? Or maybe for Elaine, there's something I've always wanted to image that I've never had a chance to put under there. Maya, anything that occurs to you there?
So I had a bit of a career shift. I studied proteins, and then now I study stem cells. And I really miss studying proteins. So I would like to go back, I think. I don't know if I'll do a PhD. I feel like I need a bit of a break because grad school during a pandemic has been quite hard. But if I did go back, I think I would do X-ray crystallography again. I did I worked in a couple labs that did that, and I ended up helping to solve like 3 protein structures, which was really exciting. I think it was one of the best things that I've ever done. And I would love to do it again. It was a lot of fun.
Nice. Elaine, what's a dream project for you?
I'm living the dream. Because I have so many different avenues coming at me to get in, the materials and stuff. Every day is a new experience because it's always the leading edge of somebody's research. So it's always new. The weirdest thing I've looked at is aerogels, but that's another story.
What is an aerogel?
Oh, it's some material that NASA uses they used it to collect dust in a comet tail. It's an insulator, so you can take a piece and you can put a Bunsen burner underneath, and a very small piece, you'll be able to touch the top and not get burned. Okay. It's 98% air mostly, which means you can hardly see it, but it's made of silicon dioxide, and but it's weird because you put it in the scanning electron microscope and the electron beam hits it and it swells up and collapses and swells up and collapses. So you go, you didn't kill this yet. It's still alive. Anyway, it's a collaboration with UVic and Triumph and the synchrotron and Japanese space group.
Fun.
But then, but the biologic, I think the diatoms are coming out to be so cool. But now slime molds, if you take spores from slime molds, they're beautiful. The sculpturing in them. And that's another story.
Vajnavi, any dream project for you?
Well, just hearing Maya and Elaine talk, I'm like, when I was in my undergrad, we worked with a lot of what we call model organisms. So a lot of organisms which are similar, like the proteins and all these different things are similar with the humans. And then I used to work with worms called C. elegans and fruit flies called Drosophila. And they were just in my internships, but I still look back and I'm like, I wish I could. I work with Saccharomyces cerevisiae here right now, so budding yeast. So you're yeast in the bread, basically. But
Okay.
I do look back and I'm like, I wish I could just return back to those dissections, those kind of microscopy and all those things. So yeah, just, I don't know, doing the same project in those organisms, which our collaborators are doing anyway. So
Very fun. We have one more question from our chat here. People were wondering, any advice on getting internships? Any suggestions if you want to work in a particular field?
Each department has internships. Each department has directed studies money and co-op students. I love the co-op students best because they're committed and they are good. And if I can get a co-op student, that'll be really good. But I'm just been asked, can I take an intern for the summer? And I'm saying, yeah, sure. Directed studies people come down. All the departments tend to get directed studies because it gives undergraduates experience in doing a project. And I often get them to train them up. You just have to keep your networking open.
Okay. Maya, any tips on finding internships?
Yeah, so in my second year, that's sort of where I started doing these directed studies. At my university, they called them, what did they call them? I don't remember. Interdisciplinary studies, that's what it was. And basically, you just have to, you build up your confidence. I faked it until I made it. Talk to your professors after class. Sit in the front row, be really engaged. University is where you want to be a keener student. 100% of the time. I sat in the front. I answered questions. I talked to my professors, my TAs after class. I went to their office hours. I talked about their careers and what they did. I emailed professors a lot. And when I was I was also in the co-op program, and after doing a couple co-ops in my university, which was in Alberta at the time, and it's very agricultural based, I'm more interested in medical and biomedical sciences. So I had emailed my current professor here at UBC for a co-op. I just out of the blue, it's called a cold call. I just set an email up and I had my co-op advisor help me send it. And I sent about maybe 6 emails and my professor got back, and now I'm here. So cold calling and just making sure that you're getting the most of your education is my recommendation, because you just need one person to say yes to you, and then that'll propel you forward.
Vajnavi, any final thoughts on that?
Yes, I completely agree with what Maya said, because like I can just give you some ideas at UBC, like the BC Children's Hospital, they have summer internships, Center for Blood Research. So there's like Elaine and Maya mentioned, there's a lot of programs here. But yes, you do have to start off by emailing profs for that. And like I completely agree with what Maya said. That's what I would do, interact with your professors. Maybe there's like tutorials, or just email them if you have some more questions about the field. That way, if there's a class of 200 people, they know who you are. Otherwise, if you're just emailing and be like, hi, I'm interested, chances are they may not like get back to you. Not because they don't know you, because these people are really busy and they get like thousands of emails in a day. And just be very like particular, not like just a general generic email. Make it like focused to what you're interested in, and like if they see you're not writing about something which is like related, then there are chances they may not pay attention to it. So just be very clear with that. And like I agree totally with what Maya and Elaine said. Just follow those steps. Yeah.
Well, thank you all for your. Oh, sorry, Elaine. One more thing to finish up.
Professors really like to talk about their research. Just have to get them talking. Like,
Yep,
Brian,
absolutely.
what's the most important thing? What do you like about your job?
Oh, I love that it's always different and new things going on. I have the title of Director of Performance and Fun Times, and days like today are fun times.
That's the definition of science.
Yeah. Thank you so much for sharing all of your experience, all of your wonderful answers to these questions. For our participants, if you're joining us at the live event today, please head back to the main website. We're going to be getting into our game show in just a moment. If you're following up afterwards on our YouTube channel, I will echo my 13-year-old daughter and say, mash that like and subscribe buttons down below. Get the notification. So we have so many mentor sessions that come up. All of the ones that happen throughout the week, we've had 30 different mentor sessions. If you've missed any of them and you want to check them out, they're all recorded. You can see them there. Thank you so much. It's been great to have you all here.
Thank you.
Thanks, everybody.
Luck.
Bye.
Bye.